All right, so we're just about on time. It's my pleasure to introduce Gareth Powell. Now, show of hands, how many of you already know Gareth Powell? Yeah, I feel kind of silly introducing him because like half of you know him. But you don't know his Beetles side, you know the other human side. So Gareth obviously did his bachelor's here, did his master's here, then moved over to Utah to Brigham Young to do his PhD, then came back east to the Florida State Collection of Arthropods as a curator, and is now at North Carolina State University as an assistant professor. So you get to see, I guess some of this might be what you started with here. Yeah, and then where he's progressed from Purdue to kind of shine for our department, which is nice. So with that, here's Gareth.
Let me dim your light. Ah, yes. See if we can put people to sleep with phylogeny. Well, thank you all for coming. Thanks, Aaron, for that introduction and the invite. It is really exciting to be back. The last time I was in this room was for my master's defense. Spoiler alert, it went okay. They did give me the degree, or at least no one else has found out that they didn't after that. But yeah, excited to talk to you today about some of the work I've been doing for the past 8 or 9 years or so. So Aaron just gave a great overview of this slide. This was kind of my journey around the country, bachelor's and master's here, with a couple years in between working at the PERC, where I got trained a lot of the basics, beetle taxonomy systematics that hopefully paid off here a little bit later. I went out to BYU for PhD and a short postdoc, where I expanded a little bit, staying with beetles, but added some feeding behavior evolution, sensory system evolution, really using beetles to answer macroevolutionary questions, more so than just working on the systematics of them. I was fortunate to get a curator position after my a short postdoc at BYU in Florida. 12.5 million specimens, 2.5 million beetles. All of those behind me are beetles. That was very hard to leave. But an opportunity came along for me to move to NC State, where I have a split appointment. I am director of the Insect Museum, similar to Aaron's position here, and then also got to start the, we called it the PI Lab, the Powell Insect Evolution Lab. And some of you over here heard me talking about it. If you see the tarsal formula of that beetle, I don't think there's any known beetle with that tarsal formula. It is 314. So that's more just for me. But in the Powell Insect Evolution Lab, we really focus on phylogenetics and using phylogenetics and phylogenomics to answer those macroevolutionary questions. So many of you are probably familiar with this Dobzhansky quote, "Nothing in biology makes sense except in the light of evolution." Steve's about to scoff. I'm going to add to this quote. It is probably not Mike Whiting. Yes, that's who said this to me the most. Nothing in evolution makes sense except in the light of phylogeny. And really, phylogeny can get at many of life's great questions. Until we had this wonderful topology here, I don't know how we would have ever known that eggs predated chickens. But really, phylogenetics provides us the tools to uncover patterns, and that those patterns are providing evidence for deciphering, in this case, the evolutionary process. It's also a method we can hang our classification on. So here you can see a nested hierarchy depicted on this phylogeny. Everything kingdom, phylum, class, order, family, genus, species can be circumscribed on a tree and tested with a tree. And then once you have a topology, however good or bad, we'll get into that here in a little bit, there are a lot of great things you can do with it. The thing we're going to focus on today is divergence time estimation. So adding the context of time to a phylogenetic estimate. This can be done at the micro scale. So here we have a few lice and their hosts. And knowing what we know about those topologies and the dates, we can see when a host shift may have occurred, what was happening in the world, what was happening with early Homo evolution when that shift took place. And maybe we can uncover something about the evolutionary history here we wouldn't have known without that context of time. We can also zoom way out. Here's a world mosquito phylogeny, came out in the last year or two. And using a divergence time, getting clade ages for all of these major radiations of mosquitoes, we can start to see where these patterns line up with tectonic plate biogeography across at the continental level. So here are the three stories I'm going to try to tell you today, and I'm going to make the case that divergence time in each case helped us answer the evolutionary question that we had. Starting with beetle bioluminescence. So not little brown beetles to start, but we'll get there, I promise. So in 2018, although kind of end of 2018, beginning of 2019, this wonderful paper, I have to stress, wonderful paper, was published in Science Advances, investigating the response of bats to aposematic signals in fireflies. It was a unique paper in that you don't probably immediately think of, let's get the mammal maybe least famous for vision to test the effect of a visual signal on. But that actually might be why this paper is so good. And even with echolocation, this paper demonstrated that the aposematic signal was the strongest in fireflies or in taxa that bioluminesce that the bats could observe. Now, I just got done telling you how good it is. They ended that paper with the following sentence. We predict that a time-calibrated firefly phylogeny will reveal that bioluminescence emerged with bats and that bats may have invented fireflies. Vertebrates get way too much credit. I mean, I'm in an ento department today, so maybe I don't have to harp on that too much. I don't know why I just found that so annoying and actually begged my advisor to let this be my postdoc to see if we could test this. Now, I will say, ignoring the underlined part, they did set us up for a wonderful spike here, right? A time-calibrated phylogeny of fireflies would certainly give us an amazing test of the origin of bioluminescence. And around that same time, so literally later that year, 2019, my lab mate happened to publish the largest firefly phylogeny to date, both in terms of taxon breadth. I think all the major subfamilies, at least at the time, Chesperitini were not on here, were sampled, and data depth. So this is a target enrichment phylogeny based on about 450 loci. I think final alignment size about 250,000 base pairs. Pretty robust phylogenetic hypothesis. I'll also mention that later on in this story, independent phylogeny was published also in related taxa. So there's a few Lampyrids here, focused more on the click beetles, but that will become relevant in a moment. The same year as well, massive year for bioluminescence, the oldest known fossil with an observable light organ was also described. So while this is not a firefly, it was described in an extinct family, the Cretophagotidae, sister to the fireflies, kind of in the clade of ragophthalmids plus phangodids, for those of you familiar with Elateriform taxonomy. But you can quite plainly see a light organ in about the right place on about the right beetle that happens to be a perfect potential calibration point for us. And so it gave us the opportunity to test this question that they ended their paper with. That's exactly what we did. This was published just a couple years ago now with some wonderful co-authors. And here we have that exact topology published by Martin, calibrated with multiple fossil calibration points, including the Cretophagotes, and then with bioluminescence reconstructed on this tree. So we won't get really deep into the methods, but another amazing thing you can do with phylogenetics is ancestral state reconstruction. Using observable traits in the tips of this tree, we can predict the likelihood of the origin and the change of those traits through time going back across the phylogeny. And that's important here. You'll see we reconstruct two separate traits, aerial bioluminescence and terrestrial bioluminescence. So about half of firefly adults bioluminesce as adults, or about half of firefly species bioluminesce as adults. Many of them around here. You're very fortunate in Indiana. We have a lot of flashing bioluminescent fireflies. Utah, there was almost none. I went 4 years without seeing one. North Carolina has a bunch though, so now it's okay.
They're on the ground.
Yes. Well, and that's the other part of this story is while about half of the adults bioluminesce, every known larva, every firefly larva, every ragophthalmid larva, every phangotid larva, all of them, without fail, bioluminesce as larvae. This is also important because when you're looking at the evolution of bioluminescence as an aposematic signal, in the larvae, it is only operating as an aposematic signal. In the adults, it's co-opted for sexual selection. There's all kinds of amazing courtship behaviors, call and answers that take place. So not all adult bioluminescence is functioning as an aposematic signal. But larvae are not mating. in larvae, this is an isolated example of apostoatism. And so here, I understand there's a phylogenetics class that will get this recording, so I'll give a little bit of a tip to any phylogenetics students in the room. If there is a fight or a debate you don't have to get into, don't get into it. So with divergence time estimation, it's kind of long been thought of as a black box. If you change the model, you're going to change your answer. If you adjust the shape of the distribution, statistical distribution you put on that calibration point, it'll change the answer, blah, blah, blah. What we decided to do to head some of that off is run every combination that was even remotely reasonable and see that all of those combinations show the same pattern. So here we did birth-death models, Yule models, that just basically takes into account extinction at different levels, had several different shapes of distribution for the calibration points. And we used the two different data sets. So that one I quickly mentioned that came out during the study, we ended up dating their topology as well. So with different taxon sampling, different probe sets, different stats, different calibration points. In all cases, you can see that terrestrial bioluminescence originated first. And then in almost all cases, we're looking at the late Jurassic, early Cretaceous, followed later, sometimes about 5 million years later, upwards of maybe 25 million years later, aerial bioluminescence popped up on the scene. But in all cases, both the adult and the larva were bioluminescent by the mid-Cretaceous, which is really important because the earliest possible even ancestor to bats didn't show up for another 50 to 75 million years right there. Now, attempting to prove someone else wrong is fun, but let's see if we can provide maybe a better alternate hypothesis. We asked ourselves what was around in the right area to have possibly driven early bioluminescent evolution. And here's the kind of rogues gallery we came up with. So all of these lineages were already diversifying by the first firefly or the first lamproids that were bioluminescing. Many of them are also associated with the right microhabitat, so kind of wet, stream adjacent, ground dwelling habitat. We have anurans, spiders, harvestmen. Most of the squamate lineages had popped up by here, ground beetles, regiviids, and then more just to prove that we did sneak a photo of a pterosaur on here, because honestly, that is the only aerial predator that we could really come up with that might have been around early enough. Now, it is grayed out for two reasons. One, we have absolutely no evidence that pterosaurs ate fireflies, right? How could you? And also, again, the story being that bioluminescence originated on the ground. So the aerial predator is not responsible for the origin of bioluminescence at all. And so with that, we would like to kind of amend their last sentence a little bit. Oh, I should mention, the person who wrote that sentence was a co-author on our paper. So we did actually get someone from that paper to help us adjust this. We predict that a time-calibrated firefly phylogeny will reveal that bioluminescence emerged before bats, and that is more likely terrestrial predators were more likely to have driven the early evolutionary history of bioluminescence in fireflies. Bats may have contributed to the maintenance of those signals much later. So there's our first example where without a divergence time, you've got very high-profile papers just spouting nonsense. Okay. So sticking with another example, here going back to my first love of little brown beetles, we're going to look at repeated feeding yield evolution and the impact of competition on diversification. So feeding behavior is a pillar of evolutionary research. I think you can say that anytime you get to cite Darwin on your slide. Shifts in feeding have been associated with changes in diversity of a clade, the morphology, behavior, biogeography of a clade. Basically, it's really important, evolutionarily speaking, because everything has to eat, at least at some life stage. And there are a couple of really classic examples within beetles. So here is the beetle tree of life at the very bottom here in red. You see a clade we refer to as Phytophaga for the non-coleopters in the room. This is leaf beetles, longhorn beetles, weevils. Basically, we'll refer to this as the most successful lineage of life within the most successful lineage of life, at least in terms of described species. And this has been a case study for looking at how feeding shift can promote diversification. Coined an adaptive zone, right? Of shifting your feeding behavior, you're now able to occupy a different niche space and resulting diversification in those lineages is pretty common. But we started to ask ourselves, and so in most of these cases, it is trying to isolate one shift, the effect of one shift, because I think that is where logically we would start. But we started to ask the questions around what happens when multiple shifts occur in a lineage, and what happens when multiple lineages shift. And around the same time, a meta-analysis came out, and it was examining trends around diversification. It tried to lump things into one of two categories. Everything's either good or bad here for this example. You had positive interactions, and these included shifts to herbivory. The phytopic example was included. Mutualisms, pollination syndromes. Basically, these are species interactions that affect one or both of the lineages in a positive way in terms of diversification. And then almost everything else is lumped as a negative interaction. So competition, parasitism, predation, most of these decrease the diversification of one, or in some cases, both of the lineages in question. And because I'm a glutton for punishment, we once again wanted to see what happens when you make it more complicated and both are happening at the exact same time. Which wins out, basically. Are the good interactions or bad interactions stronger? To ask this question, we needed a wonderful model system, choosing the Cucujoidea. Now, where's the Sylvanid student? Sensu Latu. So this is Cucujoidea plus Erotiloidea plus Nididuloidea. About 12,000 described species, 25 families, and they are cosmopolitan. But most importantly, all known feeding behaviors across Coleoptera are exhibited by at least one member of this group. Again, depending on how you group things, I'm sure Andrew, Aaron, everyone is thinking very intently right now on what's a feeding behavior a Tanib does that I've never thought of. But broadly speaking, you have a lot of the feeding diversity, in fact, exhibited by most holometabolous insects somewhere in this group. You have weird things like petal mining, right? Who's ever seen a petal miner? I didn't know there were petal miners until a few years ago. Slime mold feeding, association with cycads, things like that. And in this system, we had several objectives. We wanted to investigate the potential influence of several factors on the diversity of cucujoid clades. The age of the food resource is often cited as a driver of diversity. So if you shift to something that is older, you are likely to be more diverse. If you shift to something that is more diverse, you are likely to be more diverse. So we include diversity of the food resource. And then a factor we kind of came up with, time to colonization. So is getting there early on in the food resources evolution also important? And these are things that have exist in the literature. They're often touted as the major explainers of the diversity we see. But mainly it is to look at both a positive and negative interaction happening at the same time. So you have this first colonizer, you have this famous adaptive zone, but then you also have competition when you have subsequent lineages also shifting to the same food resource. So which wins out? To ask this question, I will go through the methods here too deeply, but we made a big tree. Almost 300 taxa, about 230 in groups. This is a little different one in that it's legacy data. And Aaron gave me a little setup here too. A few of the taxa included in this tree were based off of my master's and were generated right here in the basement of Smith Hall. And then many others from many other sources. The trick with this was we had a wonderful team of co-authors, but getting 12 coleopterists to agree on the placement of any fossil was a trick. And so we did a review of all fossil cucujoids, and I think it was like 400 taxa. These were the only 11 that all 12 of us could agree on. So we threw out 389 fossils and kept 11. And here's the result of this analysis. You can see your guys' very own Stephen Cameron made an appearance on this paper. And once again, I didn't want to get into any arguments about clock models, so we ran 3. But going forward, we stuck with log normal for the subsequent analyses here. And this is a massive tree. You don't really need to see anything on that tree other than most of the major clades of cucujoids are represented here. You'll see that most of the families are they exist by late Triassic, early Jurassic. Most of the major tribes, subfamilies kind of popping up early Cretaceous. And then really many of the extant genera you'd be familiar with today are here by the early Paleogene. Now we have a dated tree. We're able to reconstruct these feeding shifts on the topology. And I did not think that the hardest part of this study would be defining the word behavior, but it is certainly the hardest part. So when we talk about feeding behavior and the rest of this story, it's not about diet. It is not what is actually in the gut of the beetle, because that doesn't really capture the whole evolutionary history of that shift. You can go to flowers and eat pollen. You can, or I guess you could go to flowers and be scraping spores of fungi. You could go to a mushroom and scrape spores of fungi. But the selection that the flower visitor is experiencing versus the fungivore is very, very different. And so for this case, we're doing kind of the associated behavior, while in sometimes, I guess, broader context of feeding ecology surrounding acquiring food, not just what was in the gut. This was coded for the subfamily and family level and was retained when at least 80% of the known taxa exhibited the same feeding behavior. Some groups just got so complicated. I couldn't include my favorite carpophyllines because they don't do any one thing. They do a bunch of things a little bit. And then we were able to go into the literature and gather the ages for those food resources. And here they are, or at least some of them are. You can see down there in the bottom left, Myxomycota, those are slime molds. I don't know whether those were just here at the beginning of the planet. Most of the divergence times for that lineage are insane, 500 million years old. And then we go through kind of fruiting-bodied fungi, cycads. We have two origins of trees. Trees are non-monophyletic, but to get at subcortical behaviors, you have to kind of consider both trees. Flowering plants make an appearance, palms. We even have a family of cucurbites that are obligate nest parasites in birds. You can't have a nest parasite until you have nests. You can't have nests until you have nest-making birds. So that's the origin of birds. And here are color-coded all of the associated family group names for the cucurbites that are associated with each of those lineages. So here you can see we have some nice, well, nice for a macroevolutionary biologist, replicates. No one skewer me for that. But erotilines, nididulines, phylacterids, the three in red, those are the three major fungivorous clades. And I should say that's phylacrine phylacterids. We're in the process of changing it. Lamophleids, sylvanids, pachandrids, prometopines, cucugids, these are all subcortical predators. And so again, using the origin of trees to account for that feeding behavior, and so on. And then the other two data points we wanted to add here is the extant diversity of each of these beetle clades and the extant diversity of each of the food resource clades. That's not on the figure, but we have that as well. And my wife was the genius behind this part of, we had the age of the food, the age of the beetle, the number of species of the food, the number of species of the beetle. How do you look for correlations between any of these to even find what to test? And so here's that. The age of the food resource clade is significantly correlated to the age of the beetle resource. Now, again, that's previously known in the literature. Older beetles went to older food resources. Our time to colonization, unsurprisingly, was correlated with both ages because those ages were used to calculate that. So we can kind of skip that. But now these bottom two rows represent every prior hypothesis explaining the diversity of these groups. Older beetle clades are going to be more diverse. Beetle clades shifting to more diverse food resources are going to be more diverse. And some of you might already be able to see none of those were even remotely significant. Pretty disappointing. That was a rough afternoon when you think you're about to uncover some really cool pattern, and there's literally no pattern at all. Or so I thought. Again, enter Allie, the genius. And she realized that when we were looking at time, especially with respect to the food resource clade, we really were asking the question of, does getting to a food resource clade quickly benefit you? We weren't asking the question of, does getting to a specific food resource clade before anyone else benefit you? So once you group the ages and that time to colonization by the specific food resource clade, now the pattern starts to emerge. So let's walk through that. The very first group, again, we estimate to shift to fungi, erotilines, about 3,500 described species. About 20 million years later, nididulines make the shift, 1,000 species less. About 55 million years later, phylacterids show up, only 400 species. Okay, starting to get excited again. C, we have the two blue ones, Fraxinothines, Boganiids. Fraxinothines beat Boganiids there by, maybe 15 million years, 5 times as diverse. Here's my favorite one because we have 5. Lamophleids, sylvanines, pachandrids, prometopians, cucugids. You can see each of them in order: 530, 200, 110, 90, 70. I mean, this is almost, you would think I made this up, right? Now, there is a massive glaring problem, and this is how you know I didn't make it up, because here is one where the pattern is exactly reversed, or is there even a pattern anymore, right? We go from 100, a little bit later, the other half of phylacterids pop up, there's 220. In fact, the last thing to arrive there, I mean, they're all quite close, but the last thing to arrive there is the most diverse, the langurides. So what's going on with angiosperms? Why would that be the one clade that's different? Does anyone want to take a stab? I mentioned, well, what we think upset this apple cart has been mentioned in the talk so far today. Anyone have a guess? That freaking most successful lineage of life within the most successful lineage of life beat everything there by 100 million years. So if there was ever an excuse for there to be maybe a few more Olibris group phylacterids than there were Catarrhidids, it's probably because I don't think angiosperms was, at least at a broad scale, operating as an adaptive zone for these lineages, because every weevil, leaf beetle, and longhorn was already killing it in these groups. But even with that, we removed it and the pattern was even stronger. But even with that, getting to a new resource is good, but getting there first is what's better. All right, so now with the third story here, we're going to stick with feeding behavior. We're going to stick with cucurgoids, but we're going to add sensory systems. So we're going to add morphology and genetics here. I had the opportunity to contribute to this review paper looking at opsin evolution across flower visiting beetles. So opsins, I'm going to oversimplify here, but opsins are basically, they are the backbone structure that allows color vision to take place, right? And most holometabolous insects are trichromatic or tetrachromatic, so they have 3 or 4 opsins. All bees, most flies have a more complex color visual system. Beetles famously have lost one of those opsins. So you can think of most beetles operating as basically a blue colorblind guy that's walking around that doesn't have, doesn't express that third protein. But what's happened repeatedly across the beetle tree of life are that the remaining 2 opsins have duplicated, mutations have accumulated, and there's what's called subfunctionalization, where the function of the 2 that remain start to separate and they can fill in some of those gaps. There was a great paper my PhD advisor published called A Cure for the Blues. And it is, if you have enough copies of long wavelength opsins and they start to separate, that you will cover the gap that the blue opsin used to give for that sensitivity. Using this tree, so here, like I said, there's a lot of origins of UV duplication and a lot of origins of the long wavelength duplication, the 2 remaining opsins. We started to ask the question of what patterns might be emerging here. I maybe sound like a broken record. I started looking at feeding behavior. And in all cases where there was a duplication event of either long wavelength or UV, there was at least a facultative flower visitor, if not obligate associations with flowers. And I'm going to draw your attention here. So there was only 1 beetle in that study that had more than 2 UV opsins, and also only 1 beetle in that study that had more than 3 long wavelengths. And I, very lucky, they're both nitidulids. So as part of my PhD, I was traveling around the museums of the world trying to figure out nitidulids. There are a little over 4,000 described nits, and Andrew can attest they can be difficult sometimes to ID. There aren't a lot of good resources. So you go back to the origin, you go back to the holotype sitting in a Paris museum no one's looked at for 100 years, and you start to take photos so that you remember what it looks like a week later after you've left Paris, the depressing trip that is. And so I found myself with hundreds of photos of nitidulid holotypes from all around the world. And I started to see, I do also understand to everyone in the room that these are all little brown beetles, and I'm about to tell you how different they all look, and you're all going to think I'm crazy because they all look the same, but bear with me. You have to admit the size of the eyes on this thing is way different to the size of the eyes on that thing, right? Both nitidulids, both little brown guys. That's a ciliine, that's a carpophyline. But I started to see this insane variation in eye size, at least relative eye size across the lineage. And I started to ask, is there something more going on, or my eyes playing tricks on me? And we're able to publish a paper showing that in clades that are flower visiting in any way, so in association with flower, you have on average 18% larger eyes. This is done as a relative measurement, kind of a proportion of lateral headspace dedicated to eyeball. So we're amassing a data set of opsin genetics, amassing a data set of eye morphology, and amassing a data set of feeding behavior. Can we put together a test to look at the interplay of behavior, morphology, and genetics all on one tree? Well, let's find out. So this is a preliminary, everything prior to this has been published. This is the unpublished part today. This is a preliminary anchored hybrid enrichment phylogeny of nitidulids. We have most of the major clades represented here. Here are the feeding behaviors known. Now we kind of grouped based on kind of class of feeding behavior here. So we're losing a little bit of the fine scale resolution, but if you're a saprophage, regardless of the type of saprophage, we coded you as a saprophage. Predators, frugivory, fungivory, anthophagy, and then we had a few inkwines as well. And then for all of the taxa available, we have this kind of metric of relative eye size, portion of lateral headspace dedicated to eyeball. And then here are the opsin genetics for this group. A small side note, up until this point, if you wanted to understand how many opsins were in an organism, you had to do a transcriptome. Transcriptomes are expensive. We wanted to see if we could mine the opsin copies out of target enrichment data, because we can include a sample on a target enrichment run for about $80 to $85. And so these are all based on numbers mined from target enrichment data, but the 8 Ts that you see down the side, those are the 8 transcriptomes available for nitidulids. And in all cases, we recovered the exact same number. So I think the method works. Now, some of the patterns here, there's a lot of text down the left, but larger eyes were correlated with feeding behavior. We knew that already. Interestingly, Bloomberg's K-test, so this is a test to look for phylogenetic signal of a character. Opsin copy number was shown to have no phylogenetic signal. So with many characters like this, especially something considered pretty complex, multiple opsin copies is quite a complex feature. The guess is often, oh, this has happened once, due to random chance, and then it would be conserved across an entire clade. And really what you have is this group of nitidulids will have multiple opsins, this group of nitidulids will not. Well, that is not the pattern at all. Opsin copy number is clearly plastic within the group. It's being turned on and off. Those duplications are taking place in relatively short time spans. Again, we'll talk about time, but in some of these cases, different members of the same genus and even subgenus have different numbers of opsins. Long wavelength opsins were correlated with feeding behavior. UV opsins, not so much, but both were correlated when you specifically consider anthophyly, yes, no. So here what we think is going on are all these other feeding behaviors that might not be visually dominated. They're kind of muddying the water. If you look at just flower visiting, the visual system really shines there. And the next step here, this is maybe the letdown part of the talk, is we don't have a divergence time yet for this group or for this tree. And part of the reason we put a pin in that is now we have a version of that tree that Andrew saw today after lunch with 450 taxa. And instead of trying to date the one with 50, we're going to try to date the one with 450. Now, some poor student is going to have to measure the eyeballs of the other 400. Any volunteers, you let me know. We are also going to have to mine the opsin genetics out of another 400. So TBD, stay tuned for that. But really what I think this is the perfect system for, and this is what I'm going to end with and kind of argue right now, is that we have a system where clearly there's an interplay between behavioral variation, ecological shifts, morphological variation, and genetic variation. And understanding which one came first within a divergence time, we'd be able to reconstruct all of these characters and really tell the story of which happened first. Is there a shift in feeding behavior and then subsequent selection pushes morphological features, genetic diversity? Or, and this would, my bias, maybe be cooler, does the genetics duplicate or random chance, does the morphology shift? And those are actually functioning as pre-adaptations facilitating the behavioral shift. If that holds true, now we have predicted value. We can start going to measure eyeballs, measure opsins, and identify the groups that might have the highest chance of shifting. to these different types of feeding behaviors. So that's the ultimate goal. But yeah, so hopefully I've made the case today that phylogenetics is awesome, but if you add the context of time, you can make it a little bit more awesome. And the bats didn't invent anything. With that, I want to thank the newly formed Pi Lab. I have Sarah Jeffers, Sarah Aculin, Anna Parker, and Kenneth Geistart. Some of you may remember Kyle Schnepp. He has been recently hired as the curator at NC State. We're very excited to have him, a previous Purdue alum as well. You can't do this research without a lot of collaborators. As much as I'd like to, you can't get everywhere on the planet. You can't collect every beetle. You can't record what every single thing is doing. You can't measure every eyeball. And so I have a lot of people to thank that have helped with this research over the last few years. Yes, and then there is Jen and Gino and Leash. All three of those people were previous Purdue alum as well. Oh, and that's Lauren. So Lauren Widener was also here. And with that, I'll thank funding sources. You also need help paying for it, despite current situations. And with that, I'd be happy to take any questions.
Able to mine the opposite data from your targeted enrichment data? Like, what was that process? How
Yeah.
did you do that?
So anyone that's ran target enrichment, kind of step 3 is always give me a single copy ortholog and throw out everything else that's junk. So we just didn't do step 3 and then grouped everything that was a potential paralog and then started mapping them to each other. So we have enough copies from the transcriptomes of what every opsin should look like. And we had to kind of come up with some thresholds of if you're more than 3% different, you're probably more than sequencing error at that point, you're probably a different copy. So yeah, so we had to go against what every bioinformatician wanted me to, well, just for that part. And I should add that all of the opsin loci were removed from the phylogenetic reconstruction. So because we knew going in that they were all paralogs, I mean, they're inherently all useless for phylogenetic informativeness because they're all copies. Those are removed from the tree data set.
Cool. Thank you.
Anyways. Oh, Matt Dimon, hey. Do you think you could leverage this kind of opsin feeding relationship and look at odor and erg? Yes, so this is always the next question. Thank you, Matt. Of now you've done vision, can you do olfaction? Again, Allie we have conversations about this almost weekly. The issue is olfaction genetics is probably 3 orders of magnitude more complicated than visual system genetics. So I can sequence the whole phototransduction pathway, every copy of every opsin, as an afterthought on my on this target enrichment run. We don't begin to know all the classes of olfactory and gustatory genes in nitidoids. And so it's at this stage just a little too complex.
Fireflies not glowing in the wet. Is that because they're different species, because there are population differences, or like environmental?
So there are 2 species that glow in Utah. They're just high elevation, kind of hard to get to, very seasonal. They literally are bioluminescing, like we have the synchronous ones here. It's just a much larger period. They bioluminesce for a couple of weeks. Part of that was I don't think I was ever in the country for the 2 weeks that they bioluminesce my entire time in Utah. It's a hilarious story though. So BYU and the museum at the University of Utah started to do firefly research, and they involved citizen scientists, and it turns out there were fireflies. They just had to go to the top of the mountains these 2 weeks. And there were so many people in Utah that thought that coincidence was fishy, that as soon as there were 2 scientists that wanted to look at fireflies, magically now there's fireflies, that they do think that BYU released fireflies in Utah. They obviously did not, but there are a bunch of bioluminescent fireflies though in other parts of the West. So southeastern Arizona during the monsoons, there's a bunch. California has some great taxa as well. It's just kind of maybe too dry. A lot of firefly larvae, they are snail and slug predators. They like wet habitat, not a lot of wet habitat with snails running around in the high desert.
So flying vertebrate predators not known to be impactful for aerial bioluminescence. Any flying insect predators during the time that possibly be impactful on that?
So we thought about that. The majority of the flying insect predators that would have been around are all day active. So, I mean, or again, the extant remnants we have now are day active, right? So odonates, my advisor being an odonate person, much like their bat story that he wants odonates to have done everything. It's a tough case to make that the most famous day active, heat of the day, you have to collect them at noon, aerial predator, somehow across its evolutionary history shifted and used to be a really awesome nighttime predator. So yeah, we couldn't think of a lot of good aerial things. I mean, it doesn't benefit bats solve this problem with echolocation, but it's not a great time to be a visual predator flying around at night anyway, right? So I think that's kind of the main issue.
Talk about the material that you use for doing a calibration. Is that fossil material inside of amber or what kind of?
Yeah, so we try to use both compression and amber, but almost always it's amber. Compression fossils, you just can't see the features you need. People will tell you they can, and they're making it up. It is, if you describe a compression fossil and you're putting it in some extant tribe, I immediately don't believe anything else in your paper. But with amber, you can get lucky. Like I said, there are about 400. I personally looked at, I'd say about half of them, so 200 of the specimens I got to see. And in ones where I know I can see the characters, the occlusion wasn't blocked, there wasn't a bubble right in front of the mandible that I needed to see. That's kind of how we kept narrowing down our data set to these few calibration points that we could all agree on.
With regard to compression fossils, are the folks doing that just morphological features or like?
No, yeah, it's all morphology, and usually it's not even the morphology of the beetle itself anymore, right? It's mineralization that's kind of replaced it. And different things mineralize differently and preserve differently. And so I think that's why a lot of that's really, and it's also not just at that fine scale. For a nididulid, we're talking 3-4 millimeters long as your average nididulid. A compression fossil where minerals have filled in a 3 millimeter gap, I'm not seeing what I really need to see. I could barely see what I need to see on a real specimen. So.
Gareth, I'm really curious about the Opson story. So the duplication expansion in long wavelength receptors is something you see in other insect groups as well. And those long wavelength receptors are all sensitive at different wavelengths within the spectrum. Mm-hmm. So I'm kind of curious what your thoughts are as to what functions.
So back in the day, it would be you get the beetle, you attach it to an electroretinogram, flash a bunch of lights all the way across, and see when it gets excited. I don't think many people are doing that anymore. Now what we have are Drosophila colonies where you've knocked out every opsin, and can you put in one copy at a time and then test those. No one with a Drosophila colony is putting knit genes in for me just yet, but we hope to be there in the next couple of years so we can actually, what does every single one of these do? And we did have that conversation of how different even does the genetics have to be before you can even begin to think it has a different function. Because there might just be a duplication event and they're just stacked on top of each other. So we had some thresholds that we discussed in that paper, and Camilla Sharkey is the first author. This is her world, so she does a lot more of the actual physiology around visual sensitivity. But yeah.
I was curious because in mosquitoes, it's presumably associated with crepuscular activities and mating.
Yeah, and some of the things that we discussed were, so, opsin duplications, again, the UV may be separate to the long wavelength. That's increased sensitivity across part of the spectrum. We do have some day-night activity variation, but with those, what we see, maybe this is better with the morphology. So you can have more facets or you can have facets that are larger. And the pattern is usually if you need more acuity and you need to see something specific, selection is more facets. If you need to see at a weird time where it's dark, your crepuscular, your few facets just get larger. And so, yeah, I think there's also a question here of expression of a single opsin versus multiple opsins. Could you just overproduce a couple of opsins and be really good at the couple of things you can do? Or does it benefit you to have a bunch of copies and express them a little bit, and then you have sensitivity? So in odonates, they've gone off the deep end with this. They have 50, 60 copies of long wavelength and 50, 60 copies of UV. no way each of those copies actually has a different function. It's redundancy, and maybe they're expressed at different times, who knows? The visual system genetics for dragonflies has just kind of gone nuts. So yeah, basically, I don't know yet. I'll hope to figure it out. I do need the Drosophila colony though, for someone to let me do it.
Would you say that insect collection at BYU is about 2.5 million? Or is that just the beetle part of it?
It was the Beetles of Florida, was 2.5 million.
2.5 million. And I know where the best mayfly collection is. Where's the best Nidadulid collection in the United States?
In the United States, my office. Yeah, so I brought, yeah, if you need to see a Nidadulid, it used to be Andy's, but no way, I passed Andy a few years ago now. Yeah, he slowed down. So I probably have, in fact, this is when I get back from this trip, any of you in collections have heard Duke's getting rid of some things. We are absorbing Duke's forest insect collection. One of the things that comes with that are some beautifully ornate 100-year-old wood cabinets. And it is the perfect size for every one of my 100 drawers of Nidadulids to fit in my office. And that is exactly what will happen about next Tuesday. So, yeah.
Back to Kate Thompson question real quick. In Heliconis butterflies, there's research showing that different opsin patterns are actually sexually selected. So male butterflies, you see the different options than female butterflies. Do you think that perhaps that could be happening in the Nidadulids too?
So we looked at sexual selection on a bunch of different things. We saw no difference in the morphology. That was easy to measure, right? Give me a male, give me a female, or 50 of each. The eyes are about the same size in all cases, at least relatively speaking. There was some variation with body size. If the females are much larger, they have the same relative eye size, but that does mean that they have bigger eyes. Whether they're functionally different, who knows? In all of the cases, we did have, I think, 6 examples. Now we have 6 examples where we have a male and a female of this. And in fact, all 6 of those were accidents where we have having multiple of the same thing got sequenced by different people. Wasting money over here. But it allowed us to ask that. And so far we've seen no difference, the males and females in those. But that's 6, right? We have 4,500 nits, 12,000 cucojoids. There could be some sexual dimorphism.
Yeah, so to go back to the Bloomberg test and the phylogenetic signal that the opposite showed a low score, is that something where if it shows a low score, it's just showing that it maybe doesn't hold enough weight to be used as an important discrete character? I mean, would something like eye size show a high score? I guess I've just never really worked with phylogenetic signal.
Yeah, so it depends. There are tests when it's categorical and there are tests when it's continuous. So eye size being continuous, we'd have to use a different test than Bloomberg's K. But yeah, I mean, really what that's asking is, I mean, it's all evolution, but there's similarity due to common ancestry and similarity due to convergence ecological drivers. If you have a really low Bloomberg's K test, that points you in the second direction. The pattern you're seeing, if there is one, isn't usually due to common ancestry. It's probably due to some shared ecology. And in this case, yeah, the Bloomberg K was low, meaning you didn't have this one subfamily or these 2 that have the duplications. But what you had is 100% of the times you had a duplication, you exhibited a single behavior. So that test was, now that's not Bloomberg's K, so you're comparing kind of 2 different stats, but that was our argument.
Could you clarify like what made the angiosperm feeders unique in their diversification?
Yeah, so, and I don't know, this is my guess. In most of those other cases, the cucojoid lineages that shifted to them were the first things to get there at all, not just the first cucojoid, they were the first beetle. And the angiosperm radiation, or at least among the first beetles. In the angiosperm radiation, we know they were almost last to the game, right? When you've already got the whole radiation of leaf beetles, weevils, and bisids that have beat you there by 100 million years, you're pretty late to that game.
Well, let's thank Gareth. I think for those of you who didn't get a chance to see him today, other than this, I think you'll be around tomorrow, helping with insect discovery.
Yes.
So, yeah. Well,
I
thank you.
have another question.
Oh, more back. Type in, we missed.
Yes.
Matt, not across the board, but there were 1 or 2 examples where 1 or both of those lineages were endemic to a specific region, whereas the other one made them cosmopolitan. Part of that issue is you know, our tree is still a subset of a subset of a subset. You know, when there's 12,000 taxa, we don't have all the biogeographic patterns yet, so TBD.
Required. Everyone's
here.
Very good.
See, okay.